Welcome everybody for joining our session 2 today for our Sisyphus challenge. I'm going to share my screen and drive the conversation today. Put that on the screen. Hopefully I confirm you guys can see my screen. All right, great. All right, so we got a lot to cover today, but hopefully this will be a useful session. What I'd like to do today, we're at week 5, which is the midpoint of our Sisyphus challenge. And so what I'm going to do is provide a quick recap of where we've come from and where we got to today. And then we're going to march through following our current set of Odyssey standard tools to allow us to design the analysis, which we can then execute across our network starting in earnest next week. Recording, good. All right. So for the last, I guess, 2 years now, I've been using this slide as a way to describe a vision of what I'd like to see us as a community aspire towards, which specifically is about us collaborating together to engineer an open science process for generating reliable evidence. And so we have the world's largest network of databases, all standardized to the OMOP Common Data Model. And yet it's still really, really hard to execute network studies and generate results in any sort of consistent, reproducible fashion. And so what we've been working towards is the idea that a study should be able to be designed and executed by first identifying the research question and specifying what are the required inputs to that process. Then to go through what I'm outlining here is a series of a waterfall diagram with a series of steps, whereby the first step is to evaluate data fitness for use by executing our data diagnostics packages and mechanisms to determine which databases across the network could be appropriate and potentially feasible for a study. And then for the subset of databases that remain, we can move forward into phenotype development and evaluation, whereby the goal there is to make sure that we can adequately identify exposures, indications, and outcomes so that we can conduct our analyses using those definitions. And that feeds into our analysis process, whereby we want to not only design the appropriate statistical models for conducting the analysis, but be able to conduct a series of study diagnostics that will tell us whether or not the evidence that we're generating is indeed trustworthy. And only after we finish that process are we left with results that can be unblinded and interpreted, and then we can disseminate that reliable evidence to the broader stakeholder community. And so I've been pitching this idea that as a community, I'd like to see us make progress towards achieving this flow. And I'm excited about the progress we're making within the Sisyphus challenge to kind of realize this idea and really start to make progress on it. So just to recap, at the onset of the Sisyphus challenge, we identified 4 really interesting clinical research questions. We had a study focused on anti-VEGF treatments for blinding diseases and the risk of end-stage renal disease. That was actually the focus of the session 1 that was held, I guess, yesterday, your time, in the APAC region that Cindy Kai is leading. On this session today, we're going to focus on the study of fluoroquinolones and the risk of aortic aneurysm. But we also have 2 other studies that are running concurrently, looking at multiple sclerosis biologic treatments and the risk of PML, as well as looking at Skyrizi and the risk of cerebrovascular events. So we have 4 really good research questions, and we want to basically translate those questions and take them through the journey from ideas to analyses to evidence. In week 1, Mark and Chan highlighted the steps necessary for initiating a study. So at the onset, we talked about why it's important for us as a community to create a protocol and to start the process of thinking about dissemination early. We showed how to create a GitHub repository that could be a place for both collaborating on documents, but also on source code. And so we basically started up with all 4 of our studies having a place for us to collaborate and work together. In week 2, Claire, Sarah, Mui, and Jing presented about data diagnostics, this first step in the pipeline. And they introduced the database profile tool, which is a single application that any data partner can run across their data set. It produces a set of aggregate summary statistics, and those aggregate summary statistics can then be used to evaluate whether any study is potentially fit for use. And we, at this time, was when we asked data partners to begin participating in the Sisyphus Challenge and to run the database profile tool and share the results, and to allow for us to evaluate those results to determine fitness for use for each of those 4 clinical questions. And Jing showed nicely how this could actually run and how it was run, and the QVIA data as an example. We are still very much looking for more data partners. I know some of you on the call have access to some data that you haven't yet participated, but we are eager to see you participate. But to provide a little bit of extra motivation and incentive, I'll just highlight the current progress that's been made for this first stage of data diagnostics. Specifically, all of the results that I'm going to show, they're now publicly available at data.odyssey.org/data diagnostics. And here you'll see an interactive web page that you can click through to see the results for all 4 of our Sisyphus Challenge studies that you can pick. But I'll just highlight briefly what we've actually learned for all 4 studies. For the first study, the anti-VEGF study in the risk of end-stage renal disease, thus far, we have identified 15 data sources that are potentially feasible for conducting at least one of those analyses. That includes data from the US, Japan, and Taiwan. It includes both public and private claims databases and both inpatient and outpatient electronic health record data. So this will certainly be the largest study in ophthalmology that I'm aware of in the broader community, and it'd be exciting to see if there are even more data partners that can participate here. For the study that we're going to focus on today, looking at fluoroquinolones and the increased risk of aortic aneurysm, thus far, 20 databases have actually been identified as potentially feasible. This includes data from the US, UK, Belgium, Spain, France, Italy, Germany, Japan, Australia, and also covers the constellation of different types of data that we may work with, including public and private claims and inpatient and outpatient EHR data. So this is very exciting in that we're now talking about having a couple dozen data sources that are at least potentially useful for the study. And Jack and Nicole and Jung-Ho and Chan can speak more to it, but I think the largest prior study of this involved 2 databases, if I recall correctly, and I think had maybe several hundreds, maybe like 1 million exposures or something like that. But we're clearly talking about here something that's an order of magnitude larger in terms of our ability to generate an interesting answer for this question. And again, I want to encourage those of you who have access to data who have not yet participated. All you have to do is be able to run the database profile tool, and then you'd be part of this list that we can then look at to explore. And I encourage the study leads to take a look at the databases and dive in and explore more about what we see so far. Just for completeness, I'll also highlight that the study on multiple sclerosis biologics and the risk of PML, thus far we've identified 11 data sources that are potentially feasible for that study, including data in both the US, Europe, and APAC regions. And the study on Skyrizi and the risk of ischemic stroke, thus far we've identified 6 databases that can potentially contribute to this study. Currently, only US data sources seem to be viable for this study, but hopefully as more data partners contribute, we can expand that even further. So it's really exciting to see how many databases were able to be evaluated, and for us to be able to use this tool to determine which databases are potentially fit for use and which ones may not have the necessary required elements to move forward. After week 1, we moved in week 2, we moved into phenotype development and evaluation, and we heard from Asya, Anna, Gautam, Christian, Aza, JPEG, Evan, and Joel as it relates to strategies for developing phenotypes for our exposures, our indications, and our outcomes. We had a bonus session just yesterday, and Evan did a great job of thoroughly investigating how to define the outcomes of aortic aneurysm and dissection. And the output of this process really was two things. One was creating the cohorts that we may need for our analyses, and the second was being able to provide an evaluation of those cohorts to give us confidence that we understand the measurement error associated with those phenotypes, and that we know with some level of rigor that those cohorts are fit for use in moving forward with our analyses. And so at the end of that process, as it relates to the fluoroquinolone study, this is when we really had identified our target exposure of fluoroquinolone systemic exposures. We identified our comparators, which is both I'm going to mispronounce the name. What's the antibiotic there? So, go ahead.
Trimethoprim.
Oh, trimethoprim and cephalosporins were our two comparator cohorts there. I was mixing those drugs together. Thank you. And then we also were able to evaluate urinary tract infection as an indication, and then evaluate the outcome of aortic aneurysm and dissection. So given that we've had a large community coming together to basically get us to that point, where we basically are this week is we're now going to talk about week 5, which is going to be the analysis design. And it could be reasonable for you to say, why am I doing all the talking given all those other steps took so much work? And that's simply because I'm standing on the shoulders of everybody else in our community, namely all of our developers of our Odyssey Hades packages. What I'm going to be able to show you here over the course of the next 45 minutes is basically the result of our community developing a suite of tools that firmly integrate together to allow us to have a standardized analysis pipeline that I'll be able to explain how we've harmonized the inputs to that process to make it possible that we can execute one procedure. It's going to perform a whole raft of analyses and generate standardized output. So I want to acknowledge and thank Martine and Senna and Adam and JPEG and Jenna and Chris and Mark and Katie and Gautam and all the other Hades developers who have contributed specific packages. And really what I'm going to do is just reap the benefits of that hard work by showing how we can actually put all those pieces together, snap together some Lego bricks, and be able to do one composite analysis. So after today, we should have an analysis designed. That means that we've made the decisions that we need to do to move into our execution phase. And following this week, we will hear from Jenna and Jack and Senna and Chung-Soo about how to actually execute an analysis following the strategist pipeline. So Chung-Soo and Senna will navigate us through a tutorial of how to actually run the procedure next week, which we're looking forward to. Following that, George and Fan and Mitch and Nicole will walk us through how to interpret study diagnostics to understand why analyses may be considered trustworthy and why others may actually continue to be blinded because they fail associated diagnostics. And then Mark and Martine and Yong are going to discuss evidence synthesis and how we can actually pool evidence across this network of databases through a meta-analysis to generate our composite results. But ultimately, this project starts with the research question, and it's going to end with the research question. And so specifically in week 9, we expect to spend our time with interpreting the results. So that means making sure that we understand what of the remaining results that we believe are reliable evidence. What does it mean? What is the clinical implications of that? And so we'll look forward to Jack and Nicole and Chan and Jung Ho presenting and talking about the results and also helping us figure out our dissemination plan, which should end in week 9 with a publication that we're ready to submit to some journal. All right. So, for a couple of years, I've been basically saying we should follow this workflow. We're basically halfway through, and we're making good progress towards following this workflow. What I'm going to do now is actually walk through what are the standardized analyses that were currently available in the Odyssey ecosystem and how they are applicable for our fluoroquinolone study. So first of all, you're going to hear me say a lot that we're talking about standardized analytics. And today I'm going to focus specifically on standardized analytics that are currently available within the strategist pipeline. For those who missed it on Friday, we had DevCon 2023, and amongst there are several really good presentations, but one of them, if you missed it, was a presentation by Anthony Senna and Jenna Reps about strategists. So if you're interested in the technical mechanics of what we're doing and how that works, I'd encourage you to take a look through that. What I'm going to do is just be the beneficiary of the fact that this strategist pipeline exists. And for each of our standardized analytics, what I mean by that is everything I'm going to talk about has a standardized input, meaning that there are explicit design choices that can be translated into parameter inputs into analysis packages. We have a standardized procedure for execution, which basically means I can read in the inputs and execute a procedure in a standardized way. And then we have standardized outputs. And in the context of our strategist modules, what we actually have is every analysis has a defined result set that is basically, or that is a relational data model. So the packages will all execute and generate results in a comma-separated file that can be loaded into a database, which then means that we can explore those results through any graphical user interface, such as like an R-Shiny application. So whenever I talk about a standardized analytics, I want you to be thinking about, okay, if it's a standardized analytic, that means it is a standardized input and it produces a standardized output. And so the question is, well, what functions do we have available that perform that standardized approach? So currently within our strategist pipeline, we actually have modules that are available for performing characterization-based analyses. So these are anything that's associated with a descriptive statistic. We have modules for patient-level prediction. So the application of machine learning algorithms to try to predict future outcomes for patients. And then we have multiple modules for population-level effect estimation. So performing causal inference on the data sets. And so in the characterization space, you already were introduced last week to cohort diagnostics. JPEG actually showed you how cohort diagnostics can be used in an iterative form to basically support the development and evaluation rapidly and locally for a given phenotype. Here we will be using it now that we've finalized our cohorts. We will want to memorialize those by assessing the cohort diagnostics one final time for all data partners who are participating in the study. We also have a standardized analysis package for conducting cohort characterization using feature extraction. So the basic idea here would be you want to summarize baseline characteristics, demographics, comorbidities, concomitant medications. We want to be able to summarize those for each of our cohorts of interest. We have a standardized analytic for conducting incidence rates. So within a target population, what is the incidence of experiencing some outcome within some time at risk? We have a standardized analytic for conducting time to event characterization. So one may want to simply look at the distribution of when do outcomes occur in relation to exposure. And this is a standardized module that we have. And then last but not least, we have a standardized analytic module for de-challenge, re-challenge. And this is particularly relevant for anytime we're doing pharmacovigilance type work. It's quite commonplace that one useful piece of information when doing causality assessment is to understand for a given person, if they're exposed to a medication and experience an outcome, if they stop the medication, does the outcome in fact resolve? And if subsequent, in some later point in time, the person were to take the exposure again, do they actually experience the outcome again? And in pharmacovigilance space, when we see these re-challenge successes, re-challenge failures, whereby a person took the drug, experienced the outcome resolved, they stopped the drug, they took the drug again, the outcome occurred again. In many pharmacovigilance circles, that's considered pretty strong N of 1 experimental evidence that there may be a causal effect. But we can actually apply that same logic to observational data, and we have now a standardized module for doing such a thing. For patient-level prediction, this is work that many of you actually on the call have contributed to. Odyssey has one of the most robust libraries for conducting machine learning algorithms end-to-end on observational data, and that's fully been parameterized to work within our strategist pipeline. And then for population level estimation, we have two modules that are currently available. We have one that's predicated on a comparative cohort design. So this is most commonly applied in a new user framework, whereby we would use a propensity score model to balance two groups, very much trying to emulate a target trial, as Miguel Hernan would argue for. We also have a standardized analysis module for self-controlled K-series designs. So all of these standardized analytic modules are available to us. What I'm going to do over the next 40 minutes or so is describe what these modules are and why they might be applicable for our fluoroquinolone study. Ultimately, at the end of this session, it's going to be up to us as a team to decide which of these modules we actually want to use, but I'm going to hope to convince you by the end that the answer to that should be, why don't we use all of them? Because we can probably learn something interesting. And then we can, of course, finalize our protocol and write up our method section of our publication based on the final decisions that we make after today's session. Does that sound good? Jack's not smiling, so.
Yep.
All right, Chan's okay. All right, now Jack's smiling, so we're gonna keep plowing ahead. All right. So, the neat part about what our Odyssey Hades team has done is by standardizing these modules and making them work all together, we can start to see what are the common elements across our analyses. And we've really distilled this down to a pretty small set of design decisions that we actually need to make effectively for all of these standardized modules. And they're the following. We need to be able to define our target cohorts. So these are specifically what exposure we're actually interested in answering a question about. We need to identify our indications. So those are the diseases that the exposure is intended to treat. We need to be able to define our outcomes as cohorts, and those really specifically are the events that that would qualify as the things that the target of interest that we're trying to study. We need to be able to define comparator groups, and specifically in the context of a comparative cohort design, this is usually the other population that you would use as a proxy for the counterfactual. And so we want to be able to define one or more of those comparator groups anytime we are doing a comparative assessment. We need to be able to define our time at risk. So this is the span of time when we are intending to observe the outcome relative to exposure. And this is usually because there's some sort of hypothesis by which the relationship between the exposure and the outcome is based. We may have common restrictions that we want to impose in our analysis. These would be things usually like age, sex, or calendar time, which we may want to introduce in a given study. Standard Odyssey best practice that we advocate for is the use of negative controls, particularly whenever conducting causal inference studies. And so negative controls are going to be concepts that we basically are going to swap in as candidate outcomes. And the point of negative controls is for us to be able to estimate residual systematic error and perform empirical calibration. And then the last item, which is more procedural, but it's a necessary decision that we have to make, is if we're doing a comparative cohort design and using a propensity score model, we need to be able to identify which variables are not going to be included in the propensity score model because they are explicitly predictive of the exposure itself. A simple way to think about this is the drug that makes up the target and the drug that makes up the comparator, they definitely need to be excluded from a prediction of whether you're in the target or the comparator group. But sometimes we have to put in a couple extra variables in there, and I'll explain more when we get there. So this is generically and abstractly like the way that all of our standardized analytics modules work. And what's really exciting about the strategist pipeline is that even though we have all those different characterization and prediction and estimation tools, they're all now using the same set of inputs as a common structure. And so whether we are conducting the anti-VEGF study earlier today or yesterday in many of your contexts, or we're now talking about fluoroquinolone, we're going to apply the same standardized procedures. We're just going to change what the inputs are and be able to produce output in the exact same format so that, for example, interacting with the results can be done using the exact same tools. So in the case of our fluoroquinolone study, we can talk through what the design decisions are. And for purposes of this conversation, I'm gonna be sounding like I'm being assertive about what decisions have been made for the fluoroquinolone study, but I really am just posing those as the starting point for the team to be thinking about, challenging, and ultimately, at the end of this process, we want to finalize decisions that we make. And so I will just walk it through, but I encourage you all to stop me and challenge anything as I'm walking through it. So, design choices for our fluoroquinolone study. So where I think we've landed based on the draft protocol that's in our Odyssey MS Teams, as well as all the way through yesterday's conversation about the outcome, is that we have one primary target cohort, and that's fluoroquinolone systemic exposures. We have two comparators. We have trimethoprim systematic exposures and cephalosporin systematic exposures. This is all going to be nested within one indication, and that's urinary tract infections. We've identified 3 outcomes that we want to study. We want to study aortic aneurysm on its own, aortic dissection on its own, and then we will also have a composite outcome of aortic aneurysm or dissection, which is really just the combination of the two. Interesting thing that was proposed in the protocol was that there is not yet clinical consensus about the time at risk associated with fluoroquinolone exposure and the outcome, with the prior papers having a quite variable set of windows that they explored. And so it was proposed that we investigate 4 different time at risk windows: a 30-day fixed window, a 60-day fixed window, a 90-day fixed window, and a 1 year or 365-day fixed window. Whereby what those windows mean is we take the start of exposure, so think of that as the exposure of fluoroquinolones, we add a day to it, and that becomes when you become starting to have time at risk. And then we follow that up until 30 days after the start at risk. A given person may be right censored for end of observation period or what have you, but this is basically the eligible time when we will be looking for outcomes. We do have a couple restrictions. I mentioned here in the protocol, we suggested age greater than or equal to 35. You're going to see when I get to the code, I suggest that we also restrict on calendar time and specifically focus on the time prior to the COVID pandemic. So I suggest we use something like 2010, January 1st, 2010 through December 2019. And the reason for that is we did see in cohort diagnostics that patterns of use went a little crazy while people were trying to figure out what to do with COVID. So that's something to pay attention to. Negative controls is not something we've talked about at length, but there is a tool in the Odyssey community called the Common Evidence Model. From within Atlas, one can get recommendations of candidate negative controls. I've gone through the process of just clicking through the buttons to do that and creating a set of negative controls. But one explicit piece of homework that I'm going to ask the team to do is review the candidates that I've extracted out and eliminate any that you don't agree with. I've identified, I think, around 50 that I think are pretty good, but I am not a clinician and I'm not an expert in infectious diseases, so I'll defer to all of you about taking a look through those. And then the last bit is the excluded concepts. JPEG and Justin have developed a really cool Odyssey tool which supports comparator selection recommendations. And as part of that tool, one of the innovations we are soon to push out is the ability to also see what concepts may be recommended to be excluded for a given study. And I just used that to identify if there was anything that was going on. I'll give a very simple example of this, just to be more concrete about it. Let's say we're doing a comparison of fluoroquinolones versus trimethoprim. Well, since we made the decision yesterday that we're just doing trimethoprim, that means that the SMX is not actually explicitly part of the comparator choice. However, since in many databases, almost all of the trimethoprim is in combination, we would actually expect that if you allowed for a predictor to be SMX, you would see the groups totally separate. And so it's important for us to be able to identify that kind of issue up front and add it to our excluded list so that we don't inadvertently say that we can predict treatment assignment simply because the drug is being used in combination with another active ingredient. All right, so let me just stop here. Amir, you raised your hand. Great.
Yeah, I
Go
just
ahead.
want to know what is your kind of thought process when you look at negative controls? Do you start to think about the whole causal structure, whether it is similar or not similar to the outcome of interest, or you have a different thought process here?
Yeah, it's a great question, Amir. So one of the things that I hope to release this week is actually an entire hour just on the negative control process. We've done a lot of work in the Odyssey community focused on this, and where our current like standard practice that we recommend is there are others out there who think that what you should do is you should pick one or two negative controls and make sure that they have the exact confounding structure of your question of interest. We see a problem with that for two fundamental reasons. Number one is if you knew the exact confounding structure of your question of interest, you would already know the answer to your question. The whole point of confounding is that's inherently unknowable. And while you can guess at certain relationships, anybody who's drawing little cute little DAGs is oversimplifying the problem. And in our context, where we've got tens of thousands of potential covariates, and any one of them can have some degree of confounding, I think the reality of the problem is too complex to be able to model perfectly and project out. The second problem with selecting one or two negative controls, even if you believe they have the right confounding structure, is that only having one or two negative controls doesn't provide you a sufficient sample to really understand the range of systematic error that exists. And so the approach that we've been advocating for and have had good success with is to sample a relatively large collection of negative controls. Like in this case, I think I picked like 50. We've done studies with 100, with the idea that each negative control has some unknown confounding structure. It may or may not share some of the common confounding structure of the question of interest. But if you think of it as an exchangeability problem, then as long as the question of interest could have been extracted from this sample of negative controls, then we can ask ourselves the question whether or not the analysis design is able to account for whatever confounding was observed across that constellation of negative controls. And on the one side, you'd say, if we can show that across a wide range of negative controls, we don't see systematic error, we should be reassured. And on the flip side, if we saw that there is residual error, even if it's not the exact same confounding structure, we should still be worried about the design. And so what we've seen is that some designs, some epidemiologic study designs produce error in somewhat unpredictable ways that maybe post hoc one could rationalize why some things have happened, but a priori we wouldn't necessarily guess at. But the sample of negative controls provides a much stronger basis for understanding error than the one or two negative controls, even if you believe you got the DAG structure right. There was a session that was sponsored by the FDA just last month that was convened by Duke Margolis. The FDA is now investigating their own use of negative controls in all of their studies. And so Martijn and I were able to present about the Odyssey perspective as part of that conversation, which we are now collaborating with our colleagues at the FDA and EMA to support their use of this approach.
I appreciate that. But again, because I have looked at the CEM tool on Atlas, and it gives you a list of potential candidates. So how do you pick the 50? What is your metric?
Yeah, yeah. I apologize. I went on a tangent there. So the common evidence model, what it does is it actually determines what available information we know about the relationship between an exposure and outcome. Specifically, it looks at prior published literature that's in PubMed. It looks at the product label to see whether or not the issue is already labeled, like in the adverse event section. It looks at the FAIRS spontaneous data set to see whether or not there's already an existing signal. And so the basic idea is if you don't have published papers that say there's a relationship and you're not on the product label and you don't have a signal in FAIRS, then that means that there's an absence of evidence of an effect. Now, of course, absence of evidence doesn't mean evidence of absence, but we use it as a proxy to say, therefore, this is a candidate that it might be a negative control. What we then do is that produces a list. Most importantly is it throws out all the stuff where there was a possible suggestion of a relationship. It leaves us with the list of absence of evidence, and then we just do a manual review. In the case of the fluoroquinolone study, what I did was I used all antibiotics as like the set of drugs to bounce off against the common evidence model, and it returned back, I think, 148 candidate negative control conditions. I rank ordered them by prevalence across the community, and I just started marching through the list, throwing out things that were actually related to something on the label, even though it wasn't exactly the exact term, and then leaving myself with basically like the top 50 most prevalent things that I couldn't make an argument for how it could be related. But the next step that we'll have for this team is to review that list to see if maybe Jung Ho might look at the list and say, I have a reason to think that could be related. We can throw those out. And if we're left with too few, then we can, of course, always go back and add some more.
Got it. Thank you.
Yep. Song.
Hi, Patrick. So when you say you choose from prevalence in the community, are you referring to the record counts from the databases? Okay, I see you nodding
That's
there.
exactly right. Yeah, so Anna Ostropolitz ran a study, which I guess now is about 18 or 2 years ago now, but we got a lot of databases to contribute their record counts into one composite summary. And so that's kind of our proxy for how often concepts show up in our network.
All right, thanks for that.
Yeah. Any other comments or questions about these specific design choices before I plow through the modules?
I note that we do not have on-treatment time and risk in this study design, and it's reasonable because we think our hypothesis that the fluoroquinolone use for UTI will be really shortened use, and then no one will use fluoroquinolone for UTI a very long time. But I'm not sure, but it may imply that when we use the on-treatment time and risk, then we can calculate the mean duration of use of fluoroquinolone in this population. This is a new thing we've got from the on-treatment time and risk design. Yeah, but it should be noted that we do not have that design in this study.
Yeah, that's a good point, Shannon. I'll separate out two things you said because I think they're really important. So if the question is, can we just characterize the duration of exposure, then we can do that with the target cohorts we have. And in fact, we will get that out as a standard output. You don't need to do an on-treatment analysis just to get how long were you exposed. But the second point you said I think is really important is from a causal inference perspective, we don't want to do an on-treatment analysis because we don't expect that the biologic effect is only during the 3 to 5 days that you were on the exposure. And indeed, all of the prior publications were really looking at these effects being, potentially weeks after the exposure stopped. Amir, you got your hand raised?
Yeah, I mean, one idea or question was that why do we have two comparator groups? Was there a potential issue with either of them? Or because I have faced this question recently from a journal, why do you have like two? So I was wondering, why do we have two?
So I don't know if it's, I'll chime in with my immediate impression. So one of the things that we discussed as a group early on, and Jack had a nice table of it, was that there are different antibiotics that are used in different contexts. And specifically in UTI, different countries actually have different primary treatment regimens that are offered up. So we see these two as representing the primary two, I guess, between fluoroquinolones, cephalosporins, and trimethoprim. Those kind of represent the three primary modalities for outpatient urinary tract infection treatment. But there isn't necessarily a motivating reason to compel one choice versus another.
Yeah,
But I think, Amir, to your point, you're absolutely right that what the consequence of having two comparators, both of them are just serving as a proxy for the counterfactual, which means we're now going to get two answers for every given hypothesis instead of one. And we're going to be left with the question of if they're consistent, then it will be fine. And if they're inconsistent, we're going to be left with trying to reconcile what happened.
Yeah, exactly. That's kind of a risk that we are
Yep.
taking, I guess, in terms of designing. But I mean, why not saying a cohort of either or? I mean, either, trimethoprim or cephalosporins, why should we have two? Why isn't it all of them lumped into one cohort as a comparator?
So right now, so trimethoprim by itself is its own standalone ingredient. Cephalosporin is already a class which covers a whole lot of products.
Yeah.
In general, when combining products together into any sort of cohort, one is making an assumption about homogeneity of effects within that set of drugs. So at least if one believes that drugs in a class should have similar class effects, one could buy that. But the moment that you start combining classes is to presuppose that the classes themselves are homogeneous. And so in this particular case, I feel good about using two comparators from the perspective of if one of them, if they show inconsistent results, then that does raise question about the stability of the effect. And if they both show an effect, then we know that it's not due to masking that may be induced by combining them together.
Got it. And I think we have enough sample size for each of those. So
yes, sample's not going to be our problem in this study.
Yeah.
That's for sure.
Yeah. I appreciate that. Thanks.
Yeah. All right. So given that we are generally directionally good with these, what I'm going to do is I'm going to march quickly through each of the analysis modules just to introduce what it has and to frame up how these inputs that we're agreeing to here could work for the study. So first, I just want to highlight that when we talk about a target cohort, I'm kind of showing it here in blue, and an outcome cohort shown here in orange, there's actually multiple subpopulations we may be interested in. So not only are we going to be interested in the target cohort of fluoroquinolones, the outcome of aortic aneurysm, but we could also be interested in who are the people who took fluoroquinolones and did not have the event. So you can think of that as the blue that's not in this overlap region. And we're also going to be interested in the people who took fluoroquinolones and had the aortic aneurysm. So those are our exposed cases represented in the overlap region there. And for those exposed cases, we could be interested in characterizing them both in terms of what did they look like at the time that they had the exposure, and also what did they look like at the time that they had the outcome. And so, walking through that, we can think that even though we start with one target and one outcome, there are in fact 5 cohorts that we may actually want to systematically characterize. The target by itself, the outcome by itself, the target with the outcome, the target without the outcome. And so systematically, what the strategist pipeline is going to do is conduct these stratifications and provide us evidence that we can explore in this space. So core diagnostics. This is building off the good work that JPEG has been leading in our community. For all target, comparator, indication, outcome cohorts, we want to run cohort diagnostics to be able to evaluate measurement error. And as a default, we run through most of the cohort diagnostics analytics that are available. What this means for the fluoroquinolone study is that we will be producing one cohort diagnostics output that has our fluoroquinolone, trimethoprim, and cephalosporin cohorts, our UTI cohort, and all 3 of our outcome cohorts will all be in this cohort diagnostics image, so that when we're writing our manuscript. In the method section, we can say we created all the cohorts, here's their specification, and we conducted a measurement error evaluation of them using cohort diagnostic, and here's the URL out for our Shiny app that we can explore for those results. So this will all be generated as kind of a standard output. The next module is our feature characterization. The idea, and this builds off of the characterization package that Jenna Reps built, as well as the feature extraction package that Anthony Senna maintains. The idea here is that for the target, comparator, and the indication cohorts, and for all of the outcomes, we will create a sub-cohort that is the target nested within the indication, focus on the first use with a 365-day washout period, and for that cohort, so that will be new users of fluoroquinolone who have UTIs, we will look back in their history to characterize demographics, such as sex and age and race and ethnicity and index year. And we will also characterize prior conditions, drugs, procedures, measurements, devices, observations in 2 time windows, a 30-day window preceding the exposure and a 365-day window preceding exposure. What this will provide us is the basic information that one would typically want to look at in a table 1, whereby you're simply just trying to understand who are these people that are exposed to the population. And this will be a standardized output that we will have. What this will look like for our fluoroquinolone study, we will have a target cohort for fluoroquinolone, trimethoprim, with prior urinary tract infection, age greater than 35, and prior 365 days of observation. We'll also do that for the indication itself so that we can just understand whether the exposed people look fundamentally different than the entire indicated population of urinary tract infection. We'll also do this for all 3 of our outcome definitions. Oops, yeah, there we go. In our outcome definitions, we'll use our 4 time at risk windows to do that intersection activity. When we say target cohort with outcome, we will use all 4 time at risk to find those exposed cases that meet that definition. And I propose that we would just use our default set of features that we use for most of our studies as the basis for being able to characterize these populations. What this means is we're going to have a wide array of ability to select an exposure, select an outcome, and select the time at risk, and be able to see exactly what those populations look like across each of our databases who participate. The next module is incidence rates. This is building off of the cohort incidence package that Chris Knoll maintains. The idea here is that for each of our target, comparator, and indication cohorts, we can estimate the incidence of an outcome within a given time at risk. So here, again, the target cohort is going to be restricted to the indication. We're going to require that there be a prior 365 days so that we can clean out any prior events that have occurred. The outcomes will have a clean window, which basically allows us to adjust for or remove the potential for immortal time bias. Specifically, once you observe an aortic aneurysm, you are not eligible to have another aortic aneurysm for another 365 days on the basis of our phenotype that Evan reviewed for us yesterday. The neat thing about this tool is, as a default, it will produce incidence rates stratified by sex, age, and index year, following the best practices of the work that our community pioneered in supporting of the COVID vaccines with the work that Anna Ostropolitz and Jingtung Li published about the importance of us looking at incidence across these different subpopulations as a standard. What does this mean for your study for fluoroquinolones? Here we are basically saying the target cohorts will be fluoroquinolone systemic exposure, prior UTI, age 35, greater than 365 days prior observation. For that target cohort, we will estimate the incidence for aortic aneurysm dissection in the composite, and we will do it in all 4 of those time at risk windows that were summarized, and we'll stratify by age, sex, and index year to be able to understand whether that pattern is consistent. This will actually help us a good deal as it relates to understanding whether or not the incidence rate, for example, is constant across those different time risk windows, or whether or not we do indeed observe that there's a higher risk in a shorter duration of time. We then have our module for, oops, Nicole, I see your hand raised.
Oh, yeah, just a quick question about the clean window, Patrick. For each of the outcomes, is the clean window, oops, my video's not on. Is the clean window just the clean window for the specific outcome or the clean window for both outcomes? I can't remember what we decided in the cohort.
Yeah, so in the cohort, so this number that I'm typing here, 365, that's because of a decision that was made in the phenotype, whereby the phenotype says once you, or in order to qualify as an event, you cannot observe the same code within the prior 365 days. What that effectively means is once you have an event, you are immortal for 365 days forward because you're guaranteed that you won't see another event during that duration of time.
This is just in the calculation of the incidence rates. We're not talking about the actual cohort itself for the analytics.
So the cohort itself has that 365 baked into its definition. That's what allows us to find the recurrent events.
Yeah.
But because that's baked into the cohort, when we run the analysis and specifically for an incidence rate, you have to tell the incidence rate, hey, this cohort used a 365 clean window, because otherwise it wouldn't know about that's existence. So this is so that we actually clean out the immortal time that is inherent to the definition, but it's not actually exposed by the definition.
Understand, sorry, misunderstood. Yeah. Got it.
All right. So time to event. So what's interesting in the fluoroquinolone study here is that we have 4 different time windows because effectively we don't know the time to event relationship. But we have a module that is our time to event characterization. And effectively what it does is it says for every target comparator indication, just look to show when do outcomes occur in relation to the target population. And they could occur before, during, or after exposure. And so in the context of your study, you can think very simplistically, we're gonna, oops, I didn't update that slide. I apologize. There it is. Well, it's gonna be these are the answers anyway. We have fluoroquinolone, the fluoroquinolone cohort, and then we've got our outcomes. What this means is we're going to be able to see that 30-day, 60-day, 90-day, 365. For all I know, there may be a whole bunch of events even past 365 days, but we're going to be able to see when do these events occur. Why this is particularly relevant is also because we may observe that there's a good number of events that occur right before exposure. And in fact, one of the published papers that was in one of the fluoroquinolone papers that was published by Ajit Landi, had a plot of the time to event distribution and showed that there was a lot of co-occurrence that was happening, whereby there were actually tons of, there were a good number of events that basically happened in the 30 days preceding exposure. And indeed, if we think about, probably we've cleaned this up with our use of nesting in the urinary tract infection, but when we talk about hospitalization events, you know, lots of things go bad when someone's seriously infected and getting an antibiotic in the hospital. And so that can actually cause some of these outcomes to go haywire. So the time to event distribution can be useful for us to look at that. Patient level prediction. So the basic idea here would be amongst individuals who are exposed to fluoroquinolones, can we predict who's going to go on to have an aortic aneurysm? Our standard PLP framework developed by that Jenna maintains in the PLP framework and using the Cyclops package that Mark Suchard maintains allows us to take those exact same target cohorts, create features based on baseline characteristics, and then look forward to try to predict the outcome. What does this actually mean for your study? Well, we can imagine this as the target cohort is fluoroquinolone exposure. The outcomes you've got are your 3 outcomes of aortic aneurysm, dissection, and composite, and you've already enumerated your fixed window time at risks. So what we will attempt to do is train a prediction model that will allow us to say, amongst those that are exposed, who are the patients that actually go on to have the event within the time horizon. And that may be able to help us understand whether or not the event is truly something that is truly stochastic within the population, or maybe there are specific subpopulations that have higher baseline risks that we will be able to characterize. I know I've only got 4 minutes left, but I'm almost done. Our comparative cohort design, this is actually where we put the most effort into the draft protocol thus far. And I think that this is relatively straightforward as it relates to following many of the studies that have been done in the past in the Odyssey community. This is building off of the cohort method package that Martin Schumi maintains and making heavy use of the Cyclops package for training models. The basic idea is we take the target and comparator groups, we nest them within the indication, we restrict to the first exposure, we require 365 days prior observation, and then as a standard approach, we would apply a large-scale propensity score model. We can conduct one-to-one propensity score matching, apply a Cox proportional hazard model to produce a hazard ratio, and then we can use our negative controls to be able to evaluate for residual systematic error. What does this look like in the context of our study? Well, we've already enumerated that we're going to compare fluoroquinolones to 2 different comparators, trimethoprim and cephalosporin. This exact same design will work for all 3 of our outcomes, for all 4 of our time at risks. And I don't particularly see any reason why we need to be messing with the standard approach for propensity score modeling and matching that we've been using. We do need to review those negative controls, though, so I'll provide you this concept set and you'll be able to take a look at them. Last but not least is the self-controlled K-series design. The general approach, the general idea here is that a person can serve as their own control. We can nest the person time within the indication, and then we can produce a composite effect estimate for that adjusts for calendar and seasonal splines, as well as any pre-exposure time that we have. The excuse me. The self-controlled K-series design can be applied to the exact same set of target and comparator exposures. The idea here would be that we could produce an effect estimate that would allow us to understand if we observe a comparative effect between fluoroquinolones and trimethoprim of a relative risk of 1, that we don't know that that means that it tells us the drug doesn't have an effect. It may just have the same magnitude of effect as the comparator. But with a self-controlled design, if we pass diagnostics, we'll be able to evaluate whether or not that makes that is an appropriate effect estimate. So I'm going to quickly highlight. So I just went through an awful lot very quickly. And I know I only have 1 minute left, but I want to highlight how simple this actually works as it relates to the strategist pipeline. Even though we just went through a whole raft of different methods, the process for creating one standard input file is actually pretty straightforward. We basically have a single R script that will allow us to create the strategist specification. To very quickly go through it, you just list out the study analysis that you want. So here we're going to compare fluoroquinolones versus cephalosporins. These are cohort IDs. These are available on the Atlas demo site, so that they're all generally available. We specify our indication. We specify our restrictions by gender and by age. We put in our excluded concepts, which just is the variables that we want to not put into our propensity score. In our case, we have 2 target comparator combinations, fluoroquinolones versus cephalosporins and fluoroquinolones versus trimethoprim. We enumerate our outcomes. We have 3 of them: aortic aneurysm, aortic dissection, and the composite. These are, again, just cohort IDs. We specify our negative control concept set. And then we specify our time at risk windows, being that 30-day, 60-day, 90-day, 1-year window. So all I've done is I basically just parameterized the inputs that we decided on that very first slide. And what this script does, if I were to just select it and run it, is in about 5 seconds, it produces an analysis specification document that's a JSON file. This JSON file is actually relatively succinct. It has some shared resources. These are the cohort definitions that we passed into the analysis. So this reads from the Atlas instance to grab the JSON for the cohort definitions. It determines what subsets of the cohorts we need to produce in terms of that Venn diagram that I showed. it grabs all of those negative control concepts that we're going to use to produce our effect estimates. And then finally, for every module that we choose to use, it provides the specifications of the module. We can see here that we're going to run cohort generator module to instantiate all the cohorts. We're going to run the cohort diagnostics module, and you can see all of the design choices that go into that module that are enumerated here. We're going to run the characterization module, and you can see the various parameters here. While I only had to specify a small number of items, there's quite a lot of details in all these analyses. But the good news is, as a community, we've identified the various parameters and the defaults that we can use for these things. So this analysis is fully transparent and fully exposes every design decision that goes into running this analysis, while at the same time allowing us to just make the choices that we want to. This JSON file is basically where I'm going to hand this off to Chung Soo and Senna next week, and they're going to show us how, given an input of one of these JSON files, they will be able to call a single execute function that's going to read in this JSON file, run through all of those analysis modules, and generate the standardized output. So the task we have to finish up today, and I apologize that I'm a couple of minutes late, there's some homework that I have for all of you who are participating in the fluoroquinolone study. I've created a draft negative control concept set. I need you guys to review that. I'll provide the concept set ID that's in the Atlas demo version, so y'all can just take a look and see if you're okay with it. And based on today, I just walked through all the modules and tried to make an argument for why I think we should just run all of them. Of course, the current protocol doesn't reflect all of these analysis modules, in part because we hadn't yet worked through all of the details together. But what I'd like to see the team work on this week is we need to make sure that we agree to all those decisions. And assuming that we do, we want to revise the protocol to reflect the analyses we're going to perform, which is effectively to say, describe that you're going to run each of the modules and what parameters we passed into them. And then we should, I know you guys have made progress on the background section of the manuscript. Given that that should be close to in place, now we can actually draft the method section. Remembering that the method section is just going to be an enumeration of what we chose to do, and with that can be a clean break from now we'll start generating results. Next week, Chung Soo and Senna will walk through how to actually execute the package, and that's where we'll be asking those of you who have access to data to please download and grab the package and run it, and hopefully we'll demystify the process of what it actually takes to run. In the meantime, if there's any questions about any of these modules or the inputs to any of the process, don't hesitate to reach out to me. Of course, I'll be continuing to monitor the chats in the various MS Teams channels. And I'm also going to be having office hours later in the week, but I'm also happy to catch up with y'all whenever you have something. Chan, I saw you had your hand raised, so I'll stop for that.
So how can we reply for the review? How can we respond to the review of negative control outcomes? So should we some replies in the Teams, or should we revise the concept set in the Atlas?
Yeah, that's a good question. So I would suggest that it probably makes sense to make sure the conversation is taking place in the MS Teams environment.
Yeah.
I mean, it's just a list of 50 things. So maybe an easy thing to do would just be like, maybe make a Word document of the 50 things and you could throw it out on Teams, then you guys could just review them rather than clicking on them in the Atlas. Ultimately, we'll need a concept set in Atlas to read into, but while you are just reviewing and thinking about it, you may want to have a conversation on the set.
Okay, cool, thanks.
We've put them into Excel in the past, and I think those export into CSV really easily, and that might be a nice way because then each column can be a review, and then it's easy to push back into Atlas afterwards if there's stuff we want to knock out. If you want, I can pull that and do that and put it in Teams.
That'd be awesome, Evan. Thank you.
Okay.
All right. I know I went over a little bit. I apologize for that, but I hope this session was helpful in understanding how the different modules work, and hopefully got those of you who are participating in the fluoroquinolone study excited about what we're doing for fluoroquinolones, and for the rest of you just to generally understanding how network studies work in general and how we can apply the same practice to all your future study questions that you may come up with as well.